Part A, human cheek cells as viewed by light microscopy have an irregular round shape and a well-defined nucleus that takes up about 1/2 of the cell. Part B, onion skin cells, also viewed by light microscopy, are long and thin with a rectangular shape defined by a cell wall. They are about as wide as a cheek cell, but at least 5 times as long. The cell wall and nucleus are well defined in the micrograph. The onion cell nucleus is about the same size as the cheek cell nucleus. Part C. In this scanning electron micrograph of bacterial cells, the cell surface has a 3-dimensional shape. 3 of the bacteria are oval in shape. The 4th is round and has protrusions called pili. One pilus connects this bacterium to another. Figure 4. 1a. Nasal sinus cells, viewed with a light microscope. B. Onion cells, viewed with a light microscope. And C. Vibrio tasmaniensis bacterial cells, seen through a scanning electron microscope, are from very different organisms, yet all share certain basic cell structure characteristics. Credit A. Modification of work by Ed Uthman, M.D. Credit B. Modification of work by Umberto Salvignin. Credit C. Modification of work by Anthony Donofrio, William H. Fowle, Eric J. Stewart, and Kim Lewis of the Lewis Lab at Northeastern University. Scale bar data from Matt Russell. Chapter outline: 4.1 Studying cells, 4.2 Prokaryotic cells, 4.3 Eukaryotic cells, 4.4 The endomembrane system and proteins, 4.5 The cytoskeleton, 4.6 Connections between cells and cellular activities. Close your eyes and picture a brick wall. What is the wall's basic building block? It is a single brick. Like a brick wall, cells are the building blocks that make up your body. Your body has many kinds of cells, each specialized for a specific purpose. Just as we use a variety of materials to build a home, the human body is constructed from many cell types. For example, epithelial cells protect the body's surface and cover the organs and body cavities within. Bone cells help to support and protect the body. Immune system cells fight invading bacteria. Additionally, blood and blood cells carry nutrients and oxygen throughout the body while removing carbon dioxide. Each of these cell types plays a vital role during the body's growth, development, and day-to-day maintenance. In spite of their enormous variety, however, cells from all organisms, even ones as diverse as bacteria, onion, and human, share certain fundamental characteristics. Learning objectives. By the end of this section, you will be able to do the following. Describe the role of cells in organisms. Compare and contrast light microscopy and electron microscopy. Summarize cell theory. A cell is the smallest unit of a living thing. Whether comprised of one cell, like bacteria, or many cells, like a human, we call it an organism. Thus, cells are the basic building blocks of all organisms. Several cells of one kind that interconnect with each other and perform a shared function form tissues. These tissues combine to form an organ, your stomach, heart, or brain, and several organs comprise an organ system, such as the digestive system, circulatory system, or nervous system. Several systems that function together form an organism, like a human being. Here, we will examine the structure and function of cells. There are many types of cells, which scientists group into one of 2 broad categories: prokaryotic and eukaryotic. For example, we classify both animal and plant cells as eukaryotic cells, whereas we classify bacterial cells as prokaryotic. Before discussing the criteria for determining whether a cell is prokaryotic or eukaryotic, we will first examine how biologists study cells. Microscopy. Cells vary in size. With few exceptions, we cannot see individual cells with the naked eye, so scientists use microscopes. Micro equals small. Scope equals to look at, to study them. A microscope is an instrument that magnifies an object. We photograph most cells with a microscope, so we can call these images micrographs. The optics of a microscope's lenses change the image orientation that the user sees. A specimen that is right side up and facing right on the microscope slide will appear upside down and facing left when one views through a microscope, and vice versa. Similarly, if one moves the slide left while looking through the microscope, it will appear to move right, and if one moves it down, it will seem to move up. This occurs because microscopes use 2 sets of lenses to magnify the image. Because of the manner by which light travels through the lenses, this 2-lens system produces an inverted image, binocular, or dissecting microscopes, work in a similar manner, but include an additional magnification system that makes the final image appear to be upright. Light microscopes. To give you a sense of cell size, a typical human red blood cell is about 8 millionths of a meter or 8 micrometers, abbreviated as 8 μm in diameter. A pinhead is about 2/1000ths of a meter 2 in diameter. That means about 250 red blood cells could fit on a pinhead. Most student microscopes are light microscopes, figure 4. 2a. Visible light passes and bends through the lens system to enable the user to see the specimen. Light microscopes are advantageous for viewing living organisms, but since individual cells are generally transparent, their components are not distinguishable unless they are colored with special stains. Staining, however, usually kills the cells. Light microscopes that undergraduates commonly use in the laboratory magnify up to approximately 400 times. Two parameters that are important in microscopy are magnification and resolving power. Magnification is the process of enlarging an object in appearance. Resolving power is the microscope's ability to distinguish two adjacent structures as separate. The higher the resolution, the better the image's clarity and detail. When one uses oil immersion lenses to study small objects, magnification usually increases to 1,000 times. In order to gain a better understanding of cellular structure and function, scientists typically use electron microscopes. Part A. This light microscope has binocular lenses and 4 objective lenses. The sample stage is directly beneath the objective lens. The light microscope sits on a tabletop and can be easily carried. Part B. The electron microscope shown here sits in a museum. It is about the size of a desk, and a person can sit in front of it to operate it. A column taller than a person rises from the center of the scope. Figure 4.2. A. Most light microscopes in a college biology lab can magnify cells up to approximately 400 times and have a resolution of about 200 nanometers. B. Electron microscopes provide a much higher magnification, 100,000x, and have a resolution of 50 picometers. Credit a modification of work by GCG/Wikimedia Commons. Credit B. Modification of work by Evan Bench. Electron microscopes. In contrast to light microscopes, electron microscopes, figure 4. 2b, use a beam of electrons instead of a beam of light. Not only does this allow for higher magnification, and thus more detail, figure 4.3, it also provides higher resolving power. The method to prepare the specimen for viewing with an electron microscope kills the specimen. Electrons have short wavelengths, shorter than photons, that move best in a vacuum, so we cannot view living cells with an electron microscope. In a scanning electron microscope, a beam of electrons moves back and forth across a cell's surface, creating details of cell surface characteristics. In a transmission electron microscope, the electron beam penetrates the cell and provides details of a cell's internal structures. As you might imagine, electron microscopes are significantly more bulky and expensive than light microscopes. Part A, salmonella through a light microscope appear as tiny purple dots. Part B, in this scanning electron micrograph, bacteria appear as three-dimensional ovals. The human cells are much larger with a complex, folded appearance. Some of the bacteria lie on the surface of the human cells, and some are squeezed between them. Figure 4. 3a, these salmonella bacteria appear as tiny purple dots when viewed with a light microscope. B, this scanning electron microscope micrograph shows salmonella bacteria in red invading human cells, yellow. Even though subfigure B shows a different salmonella specimen than subfigure A, you can still observe the comparative increase in magnification and detail. Credit A. Modification of work by CDC Armed Forces Institute of Pathology, Charles N. Farmer, Rocky Mountain Laboratories. Credit B. Modification of work by NIAID, NIH. Scalebar data from Matt Russell. Link to learning. For another perspective on cell size, try the How Big Interactive at _thighs site and underline. Cell theory. The microscopes we use today are far more complex than those that Dutch shopkeeper Antony van Leeuwenhoek used in the 1600s. Skilled in crafting lenses, van Leeuwenhoek observed the movements of single-celled organisms, which he collectively termed animalcules. In the 1665 publication Micrographia, Experimental scientist Robert Hooke coined the term cell for the box-like structures he observed when viewing cork tissue through a lens. In the 1670s, Van Leeuwenhoek discovered bacteria and protozoa. Later advances in lenses, microscope construction, and staining techniques enabled other scientists to see some components inside cells. By the late 1830s, botanist Matthias Schleiden and zoologist Theodor Schwann were studying tissues and proposed the unified cell theory, which states that one or more cells comprise all living things. The cell is the basic unit of life, and new cells arise from existing cells. Rudolf Virchow later made important contributions to this theory. Career connection. Cytotechnologist. Have you ever heard of a medical test called a pap smear, figure 4.4? In this test, a doctor takes a small sample of cells from the patient's uterine cervix and sends it to a medical lab where a cytotechnologist stains the cells and examines them for any changes that could indicate cervical cancer or a microbial infection. Cytotechnologists, cyto equals cell, are professionals who study cells via microscopic examinations and other laboratory tests. They are trained to determine which cellular changes are within normal limits and which are abnormal. Their focus is not limited to cervical cells. They study cellular specimens that come from all organs. When they notice abnormalities, they consult a pathologist, a medical doctor who interprets and diagnoses changes that disease in body tissue and fluids cause. Cytotechnologists play a vital role in saving people's lives. When doctors discover abnormalities early, a patient's treatment can begin sooner, which usually increases the chances of a successful outcome. Both normal cells and cells infected with HPV have an irregular, round shape and a well-defined nucleus. Infected cells, however, are 2 to 3 times as large as uninfected cells, and some have 2 nuclei. Figure 4. For these uterine cervix cells, viewed through a light microscope, are from a pap smear. Normal cells are on the left. The cells on the right are infected with human papillomavirus, HPV. Notice that the infected cells are larger. Also, 2 of these cells each have 2 nuclei instead of 1, the normal number. Credit: modification of work by Ed Uthman, MD, scale bar data from Matt Russell. Learning objectives. By the end of this section, you will be able to do the following. Name examples of prokaryotic and eukaryotic organisms. Compare and contrast prokaryotic and eukaryotic cells. Describe the relative sizes of different cells. Explain why cells must be small. Cells fall into one of 2 broad categories, prokaryotic and eukaryotic. We classify only the predominantly single-celled organisms, bacteria and archaea, as prokaryotes, before, kary equals nucleus. Animal cells, plants, fungi, and protists are all eukaryotes, EU equals true. Components of prokaryotic cells. All cells share 4 common components. 1. A plasma membrane, an outer covering that separates the cell's interior from its surrounding environment. 2. Cytoplasm, consisting of a jelly-like cytosol within the cell in which there are other cellular components. 3. DNA, the cell's genetic material. And 4. Ribosomes, which synthesize proteins. However, prokaryotes differ from eukaryotic cells in several ways. A prokaryote is a simple, mostly single-celled, unicellular organism that lacks a nucleus or any other membrane-bound organelle. We will shortly come to see that this is significantly different in eukaryotes. Prokaryotic DNA is in the cell's central part, the nucleoid, figure 4.5. In this illustration, the prokaryotic cell has an oval shape. The circular chromosome is concentrated in a region called the nucleoid. The fluid inside the cell is called the cytoplasm. Ribosomes, depicted as small circles, float in the cytoplasm. The cytoplasm is encased by a plasma membrane, which in turn is encased by a cell wall. A capsule surrounds the cell wall. The bacterium depicted has a flagellum protruding from one narrow end. Pili are small protrusions that project from the capsule in all directions. Figure 4.5. This figure shows the generalized structure of a prokaryotic cell. All prokaryotes have chromosomal DNA localized in a nucleoid, ribosomes, a cell membrane, and a cell wall. The other structures shown are present in some, but not all, bacteria. Most bacteria have a peptidoglycan cell wall, and many have a polysaccharide capsule, figure 4.5. The cell wall acts as an extra layer of protection, helps the cell maintain its shape, and prevents dehydration. The capsule enables the cell to attach to surfaces in its environment. Some prokaryotes have flagella, pili, or fimbriae. Flagella are used for locomotion. Pili exchange genetic material during conjugation, the process by which one bacterium transfers genetic material to another through direct contact. Bacteria use fimbriae to attach to host cell. Career connection. Microbiologist. The most effective action anyone can take to prevent the spread of contagious illnesses is to wash their hands. Why? Because microbes, organisms so tiny that they can only be seen with microscopes, are ubiquitous. They live on doorknobs, money, your hands, and many other surfaces. If someone sneezes into his hand and touches a doorknob, and afterwards you touch that same doorknob, the microbes from the sneezer's mucus are now on your hands. If you touch your hands to your mouth, nose, or eyes, those microbes can enter your body and could make you sick. However, not all microbes, also called microorganisms, cause disease. Most are actually beneficial. You have microbes in your gut that make vitamin K. Other microorganisms are used to ferment beer and wine. Microbiologists are scientists who study microbes. Microbiologists can pursue a number of careers. Not only do they work in the food industry, they are also employed in the veterinary and medical fields. They can work in the pharmaceutical sector, serving key roles in research and development by identifying new antibiotic sources that can treat bacterial infections. Environmental microbiologists may look for new ways to use specially selected or genetically engineered microbes to remove pollutants from soil or groundwater, as well as hazardous elements from contaminated sites. We call using these microbes bioremediation technologies. Microbiologists can also work in the bioinformatics field, providing specialized knowledge and insight for designing, developing, and optimizing specificity of computer models of, for example, bacterial epidemics. Cell size. At 0,1 to 50 μm in diameter, prokaryotic cells are significantly smaller than eukaryotic cells, which have diameters ranging from 10 to 100 μm. Figure 4.6. The prokaryote's small size allows ions and organic molecules that enter them to quickly diffuse to other parts of the cell. Similarly, any wastes produced within a prokaryotic cell can quickly diffuse. This is not the case in eukaryotic cells, which have developed different structural adaptations to enhance intracellular transport. Relative sizes on a logarithmic scale from 0. 1 nm to 1 m are shown. Objects are shown from smallest to largest. The smallest object shown, an atom, is about 1 nm in size. The next largest objects shown are lipids and proteins. These molecules are between 1 and 10 nm. A flu virus is just under 100 nm. Bacteria and mitochondria are both about 1 Greek μm plant, and animal cells are both between 10 and 100 Greek μm. A human egg is between 100 Greek μm and 1 mm. A frog egg is about 1 mm. A chicken egg and an ostrich egg are both between 10 and 100 mm, but an ostrich egg is larger. For comparison, a human is approximately 1 m tall. Appropriate instruments to view different size ranges are also shown. An electron microscope is used for things from less than 1 nm to almost 100 Greek μm. A light microscope is used for things from 100 nm to over 1 mm. The naked eye is used for things 100 Greek μm and up. Figure 4.6. This figure shows relative sizes of microbes on a logarithmic scale. Recall that each unit of increase in a logarithmic scale represents a 10-fold increase in the quantity measured. Small size, in general, is necessary for all cells, whether prokaryotic or eukaryotic. Let's examine why that is so. First, we'll consider the area and volume of a typical cell. Not all cells are spherical in shape, but most tend to approximate a sphere. You may remember from your high school geometry course that the formula for the surface area of a sphere is 4 pi/2, while the formula for its volume is 4 pi/3/3. Thus, as the radius of a cell increases, its surface area increases as the square of its radius, but its volume increases as the cube of its radius, much more rapidly. Therefore, as a cell increases in size, its surface area to volume ratio decreases. This same principle would apply if the cell had a cube shape, figure 4-7. If the cell grows too large, the plasma membrane will not have sufficient surface area to support the rate of diffusion required for the increased volume. In other words, as a cell grows, it becomes less efficient. One way to become more efficient is to divide. Other ways are to increase surface area by foldings of the cell membrane, become flat or thin and elongated, or develop organelles that perform specific tasks. These adaptations lead to developing more sophisticated cells, which we call eukaryotic cells. Visual connection. On the left, a sphere 1 min diameter is encased in a box of the same width. On the right, the same sphere is encased in a box 2 min diameter. Figure 4-7 notice that as a cell increases in size, its surface area to volume ratio decreases. When there is insufficient surface area to support a cell's increasing volume, a cell will either divide or die. The cube-like cell on the left has a volume of 1:3 and a surface area of 6:2, with a surface area to volume ratio of 6:1. Whereas, the cube-like cell on the right has a volume of 8:3 and a surface area of 24:2, with a surface area to volume ratio of 3:1. Prokaryotic cells are much smaller than eukaryotic cells. What advantages might small cell size confer on a cell? What advantages might large cell size have? Learning objectives. By the end of this section, you will be able to do the following. Describe the structure of eukaryotic cells. Compare animal cells with plant cells. State the role of the plasma membrane. Summarize the functions of the major cell organelles. Have you ever heard the phrase, form follows function? It's a philosophy that many industries follow. In architecture, this means that buildings should be constructed to support the activities that will be carried out inside them. For example, a skyscraper should include several elevator banks. A hospital should have its emergency room easily accessible. Our natural world also utilizes the principle of form following function, especially in cell biology, and this will become clear as we explore eukaryotic cells, figure 4.8. Unlike prokaryotic cells, eukaryotic cells have 1. A membrane-bound nucleus, 2. Numerous membrane-bound organelles such as the endoplasmic reticulum, Golgi apparatus, chloroplasts, mitochondria, and others, and 3. Several rod-shaped chromosomes. Because a membrane surrounds eukaryotic cells' nucleus, it has a true nucleus. The word organelle means little organ, and, as we already mentioned, organelles have specialized cellular functions, just as your body's organs have specialized functions. At this point, it should be clear to you that eukaryotic cells have a more complex structure than prokaryotic cells. Organelles allow different functions to be compartmentalized in different areas of the cell. Before turning to organelles, let's first examine two important components of the cell, the plasma membrane and the cytoplasm. Visual connection. Part A. This illustration shows a typical eukaryotic animal cell, which is egg-shaped. The fluid inside the cell is called the cytoplasm, and the cell is surrounded by a cell membrane. The nucleus takes up about 1/2 the width of the cell. Inside the nucleus is the chromatin, which is composed of DNA and associated proteins. A region of the chromatin is condensed into the nucleolus, a structure where ribosomes are synthesized. The nucleus is encased in a nuclear envelope, which is perforated by protein-lined pores that allow entry of material into the nucleus. The nucleus is surrounded by the rough and smooth endoplasmic reticulum, or ER. The smooth ER is the site of lipid synthesis. The rough ER has embedded ribosomes that give it a bumpy appearance. It synthesizes membrane and secretory proteins. In addition to the ER, many other organelles float inside the cytoplasm. These include the Golgi apparatus, which modifies proteins and lipids synthesized in the ER. The Golgi apparatus is made of layers of flat membranes. Mitochondria, which produce food for the cell, have an outer membrane and a highly folded inner membrane. Other, smaller organelles include peroxisomes that house oxidative metabolism, lysosomes that digest food, and vacuoles. Ribosomes, responsible for protein synthesis, also float freely in the cytoplasm and are depicted as small dots. The last cellular component shown is the cytoskeleton, which has 4 different types of components: microfilaments, intermediate filaments, microtubules, and centrosomes. Microfilaments are fibrous proteins that line the cell membrane and make up the cellular cortex. Intermediate filaments are fibrous proteins that hold organelles in place. Microtubules form the mitotic spindle and maintain cell shape. Centrosomes are made of two tubular structures at right angles to one another. They form the microtubule organizing center. Part B. This illustration depicts a typical eukaryotic plant cell. The nucleus of a plant cell contains chromatin and a nucleolus, the same as an animal cell. Other structures that the plant cell has in common with the animal cell include rough and smooth endoplasmic reticulum, the Golgi apparatus, mitochondria, peroxisomes, and ribosomes. The fluid inside the plant cell is called the cytoplasm, just as it is in an animal cell. The plant cell has 3 of the 4 cytoskeletal components found in animal cells: microtubules, intermediate filaments, and microfilaments. Plant cells do not have centrosomes. Plant cells have 4 structures not found in animal cells: chloroplasts, plastids, a central vacuole, and a cell wall. Chloroplasts are responsible for photosynthesis. They have an outer membrane, an inner membrane, and stack of membranes inside the inner membrane. The central vacuole is a very large, fluid-filled structure that maintains pressure against the cell wall. Plastids store pigments. The cell wall is outside the cell membrane. Figure 4. 8. These figures show the major organelles and other cell components of A, a typical animal cell, and B, a typical eukaryotic plant cell. The plant cell has a cell wall, chloroplasts, plastids, and a central vacuole, structures not in animal cells. Most cells do not have lysosomes or centrosomes. If the nucleolus were not able to carry out its function, what other cellular organelles would be affected? The plasma membrane. Like prokaryotes, eukaryotic cells have a plasma membrane, figure 4. 9. A phospholipid bilayer with embedded proteins that separates the internal contents of the cell from its surrounding environment. A phospholipid is a lipid molecule with 2 fatty acid chains and a phosphate-containing group. The plasma membrane controls the passage of organic molecules, ions, water, and oxygen into and out of the cell. Wastes, such as carbon dioxide and ammonia, also leave the cell by passing through the plasma membrane. The plasma membrane is composed of a phospholipid bilayer. In the bilayer, the 2 long hydrophobic tails of phospholipids face toward the center, and the hydrophilic head group faces the exterior. Integral membrane proteins and protein channels span the entire bilayer. Protein channels have a pore in the middle. Peripheral membrane proteins sit on the surface of the phospholipids and are associated with the phospholipid head groups. On the exterior side of the membrane, carbohydrates are attached to certain proteins and lipids. Filaments of the cytoskeleton line the interior of the membrane. Figure 4. 9. The eukaryotic plasma membrane is a phospholipid bilayer with proteins and cholesterol embedded in it. The plasma membranes of cells that specialize in absorption fold into finger-like projections that we call microvilli, singular equals microvillus. Figure 4.10. Such cells typically line the small intestine, the organ that absorbs nutrients from digested food. This is an excellent example of form-following function. People with celiac disease have an immune response to gluten, which is a protein in wheat, barley, and rye. The immune response damages microvilli, and thus, afflicted individuals cannot absorb nutrients. This leads to malnutrition, cramping, and diarrhea. Patients suffering from celiac disease must follow a gluten-free diet. The left part of this figure is a transmission electron micrograph of microvilli, which appear as long, slender stalks extending from the plasma membrane. The right side illustrates cells containing microvilli. The microvilli cover the surface of the cell facing the interior of the small intestine. Figure 4. 10. Microvilli, as they appear on cells lining the small intestine, increase the surface area available for absorption. These microvilli are only on the area of the plasma membrane that faces the cavity from which substances will be absorbed. Credit, micrograph, modification of work by Louisa Howard. The cytoplasm. The cytoplasm is the cell's entire region between the plasma membrane and the nuclear envelope, a structure we will discuss shortly. It is comprised of organelles suspended in the gel-like cytosol, the cytoskeleton, and various chemicals, figure 4. 8. Even though the cytoplasm consists of 70 to 80% water, it has a semi-solid consistency, which comes from the proteins within it. However, proteins are not the only organic molecules in the cytoplasm. Glucose and other simple sugars, polysaccharides, amino acids, nucleic acids, fatty acids, and derivatives of glycerol are also there. Ions of sodium, potassium, calcium, and many other elements also dissolve in the cytoplasm. Many metabolic reactions, including protein synthesis, take place in the cytoplasm. The nucleus. Typically, the nucleus is the most prominent organelle in a cell, figure 4. 8. The nucleus, plural equals nuclei, houses the cell's DNA and directs the synthesis of ribosomes and proteins. Let's look at it in more detail, figure 411. The two-dimensional image depicts the nucleus of a cell as a circular object with two membranes. Several gaps appear in the circle, representing nuclear pores. Surrounding the nucleus are membranous sacs representing the endoplasmic reticulum. Inside the nucleus is another circle, approximately 10% of the total size of the nucleus, representing the nucleolus. Figure 4. 11. The nucleus stores chromatin, DNA plus proteins, in a gel-like substance called the nucleoplasm. The nucleolus is a condensed chromatin region where ribosome synthesis occurs. We call the nucleus boundary the nuclear envelope. It consists of two phospholipid bilayers, an outer and an inner membrane. The nuclear membrane is continuous with the endoplasmic reticulum. Nuclear pores allow substances to enter and exit the nucleus. The nuclear envelope. The nuclear envelope is a double membrane structure that constitutes the nucleus' outermost portion. Figure 411. Both the nuclear envelope's inner and outer membranes are phospholipid bilayers. The nuclear envelope is punctuated with pores that control the passage of ions, molecules, and RNA between the nucleoplasm and cytoplasm. The nucleoplasm is the semi-solid fluid inside the nucleus, where we find the chromatin and the nucleolus. Chromatin and chromosomes. To understand chromatin, it is helpful to first explore chromosomes, structures within the nucleus that are made up of DNA, the hereditary material. You may remember that in prokaryotes, DNA is organized into a single circular chromosome. In eukaryotes, chromosomes are linear structures. Every eukaryotic species has a specific number of chromosomes in the nucleus of each cell. For example, in humans, the chromosome number is 46, while in fruit flies, it is 8. Chromosomes are only visible and distinguishable from one another when the cell is getting ready to divide. When the cell is in the growth and maintenance phases of its life cycle, proteins attach to chromosomes, and they resemble an unwound, jumbled bunch of threads. We call these unwound protein-chromosome complexes chromatin, figure 4. 12. Chromatin describes the material that makes up the chromosomes both when condensed and decondensed. Part A. In this illustration, DNA tightly coiled into two thick cylinders is shown in the upper right. A close-up shows how the DNA is coiled around proteins called histones. Part B. This image shows paired chromosomes. The chromosomes are shown as a collection of slender tubes. Figure 4. 12a. This image shows various levels of chromatin's organization, DNA and protein. B. This image shows paired chromosomes. Credit B. Modification of work by NIH. Scalebar data from Matt Russell. The nucleolus. We already know that the nucleus directs the synthesis of ribosomes, but how does it do this? Some chromosomes have sections of DNA that encode ribosomal RNA. A darkly staining area within the nucleus called the nucleolus, plural equals nucleoli, aggregates the ribosomal RNA with associated proteins to assemble the ribosomal subunits that are then transported out through the pores in the nuclear envelope to the cytoplasm. Ribosomes. Ribosomes are the cellular structures responsible for protein synthesis. When we view them through an electron microscope, ribosomes appear either as clusters, polyribosomes, or single, tiny dots that float freely in the cytoplasm. They may be attached to the plasma membrane's cytoplasmic side or the endoplasmic reticulum's cytoplasmic side and the nuclear envelope's outer membrane, figure 4. 8. Electron microscopy shows us that ribosomes, which are large protein and RNA complexes, consist of two subunits, large and small, figure 413. Ribosomes receive their orders for protein synthesis from the nucleus, where the DNA transcribes into messenger RNA, mRNA. The mRNA travels to the ribosomes, which translate the code provided by the sequence of the nitrogenous bases in the mRNA into a specific order of amino acids in a protein. Amino acids are the building blocks of proteins. The ribosome consists of a small subunit and a large subunit, which is about 3 times as big as the small one. The large subunit sits on top of the small one. A chain of mRNA threads between the large and small subunits. A protein chain extends from the top of the large subunit. Figure 4.13, a large subunit top and a small subunit bottom, comprise ribosomes. During protein synthesis, ribosomes assemble amino acids into proteins. Because protein synthesis is an essential function of all cells, including enzymes, hormones, antibodies, pigments, structural components, and surface receptors, there are ribosomes in practically every cell. Ribosomes are particularly abundant in cells that synthesize large amounts of protein. For example, the pancreas is responsible for creating several digestive enzymes, and the cells that produce these enzymes contain many ribosomes. Thus, we see another example of form-following function, mitochondria. Scientists often call mitochondria, singular equals mitochondrion, powerhouses or energy factories of both plant and animal cells because they are responsible for making adenosine triphosphate, ATP, the cell's main energy-carrying molecule. ATP represents the cell's short-term stored energy. Cellular respiration is the process of making ATP using the chemical energy in glucose and other nutrients. In mitochondria, this process uses oxygen and produces carbon dioxide as a waste product. In fact, the carbon dioxide that you exhale with every breath comes from the cellular reactions that produce carbon dioxide as a byproduct. In keeping with our theme of form-following function, it is important to point out that muscle cells have a very high concentration of mitochondria that produce ATP. Your muscle cells need considerable energy to keep your body moving. When your cells don't get enough oxygen, they do not make much ATP. Instead, producing lactic acid accompanies the small amount of ATP they make in the absence of oxygen. Mitochondria are oval-shaped, double-membrane organelles, figure 4.14, that have their own ribosomes and DNA. Each membrane is a phospholipid bilayer embedded with proteins. The inner layer has folds called cristae. We call the area surrounded by the folds the mitochondrial matrix. The cristae and the matrix have different roles in cellular respiration. This transmission electron micrograph of a mitochondrion shows an oval outer membrane and an inner membrane with many folds called cristae. Inside the inner membrane is a space called the mitochondrial matrix. Figure 4.14, this electron micrograph shows a mitochondrion through an electron microscope. This organelle has an outer membrane and an inner membrane. The inner membrane contains folds, called cristae, which increase its surface area. We call the space between the two membranes the intermembrane space, and the space inside the inner membrane the mitochondrial matrix. ATP synthesis takes place on the inner membrane. Credit: modification of work by Matthew Britton, scale bar data from Matt Russell. Peroxisomes. Peroxisomes are small, round organelles enclosed by single membranes. They carry out oxidation reactions that break down fatty acids and amino acids. They also detoxify many poisons that may enter the body. Many of these oxidation reactions release hydrogen peroxide, H2O2, which would be damaging to cells. However, when these reactions are confined to peroxisomes, enzymes safely break down the H2O2 into oxygen and water. For example, peroxisomes in liver cells detoxify alcohol. Glyoxysomes, which are specialized peroxisomes in plants, are responsible for converting stored fats into sugars. Plant cells contain many different types of peroxisomes that play a role in metabolism, pathogen defense, and stress response, to mention a few. Vesicles and vacuoles. Vesicles and vacuoles are membrane-bound sacs that function in storage and transport. Other than the fact that vacuoles are somewhat larger than vesicles, there is a very subtle distinction between them. Vesicle membranes can fuse with either the plasma membrane or other membrane systems within the cell. Additionally, some agents such as enzymes within plant vacuoles break down macromolecules. The vacuole's membrane does not fuse with the membranes of other cellular components. Animal cells versus plant cells. At this point, you know that each eukaryotic cell has a plasma membrane, cytoplasm, a nucleus, ribosomes, mitochondria, peroxisomes, and in some, vacuoles. But there are some striking differences between animal and plant cells. While both animal and plant cells have microtubule organizing centers, MTOCs, animal cells also have centrioles associated with the MTOC, a complex we call the centrosome. Animal cells each have a centrosome and lysosomes, whereas most plant cells do not. Plant cells have a cell wall, chloroplasts and other specialized plastids, and a large central vacuole, whereas animal cells do not. The centrosome. The centrosome is a microtubule organizing center found near the nuclei of animal cells. It contains a pair of centrioles, two structures that lie perpendicular to each other, figure 4.15. Each centriole is a cylinder of 9 triplets of microtubules. The image depicts two tube-like structures, one on top of the other, at right angles. Each of the tubes is labeled as the centriole. Each tube is composed of smaller tubes grouped in threes. These are labeled microtubule triplet. Each centriole tube is composed of 9 triplets arranged to form the wall of the tube. Figure 4.15. The centrosome consists of 2 centrioles that lie at right angles to each other. Each centriole is a cylinder comprised of 9 triplets of microtubules. Non-tubulin proteins, indicated by the green lines, hold the microtubule triplets together. The centrosome, the organelle where all microtubules originate, replicates itself before a cell divides, and the centrioles appear to have some role in pulling the duplicated chromosomes to opposite ends of the dividing cell. However, the centriole's exact function in cell division isn't clear, because cells that have had the centrosome removed can still divide, and plant cells, which lack centrosomes, are capable of cell division. Lysosomes. Animal cells have another set of organelles that most plant cells do not, lysosomes. The lysosomes are the cell's garbage disposal. In plant cells, the digestive processes take place in vacuoles. Enzymes within the lysosomes aid in breaking down proteins, polysaccharides, lipids, nucleic acids, and even worn-out organelles. These enzymes are active at a much lower pH than the cytoplasms. Therefore, the pH within lysosomes is more acidic than the cytoplasm's pH. Many reactions that take place in the cytoplasm could not occur at a low pH. So again, the advantage of compartmentalizing the eukaryotic cell into organelles is apparent. The cell wall. If you examine figure 4.8, the plant cell diagram, you will see a structure external to the plasma membrane. This is the cell wall, a rigid covering that protects the cell, provides structural support, and gives shape to the cell. Fungal and some protistan cells also have cell walls. While the prokaryotic cell wall's chief component is peptidoglycan, the major organic molecule in the plant, and some protists, cell wall is cellulose, figure 4.16, a polysaccharide comprised of glucose units. Have you ever noticed that when you bite into a raw vegetable, like celery, it crunches? That's because you are tearing the celery cell's rigid cell walls with your teeth. This illustration shows 3 glucose subunits that are attached together. Dashed lines at each end indicate that many more subunits make up an entire cellulose fiber. Each glucose subunit is a closed ring composed of carbon, hydrogen, and oxygen atoms. Figure 4.16. Cellulose is a long chain of beta-glucose molecules connected by a 1,4 linkage. The dashed lines at each end of the figure indicate a series of many more glucose units. The size of the page makes it impossible to portray an entire cellulose molecule. Chloroplasts. Like the mitochondria, chloroplasts have their own DNA and ribosomes, but chloroplasts have an entirely different function. Chloroplasts are plant cell organelles that carry out photosynthesis. Photosynthesis is the series of reactions that use carbon dioxide, water, and light energy to make glucose and oxygen. This is a major difference between plants and animals. Plants, autotrophs, are able to make their own food, like sugars used in cellular respiration to provide ATP energy generated in the plant mitochondria. Animals, heterotrophs, must ingest their food. Like mitochondria, Chloroplasts have outer and inner membranes, but within the space enclosed by a chloroplast's inner membrane is a set of interconnected and stacked fluid-filled membrane sacs we call thylakoids, figure 4. 17. Each thylakoid stack is a granum, plural equals grana. We call the fluid enclosed by the inner membrane that surrounds the grana the stroma. This illustration shows a chloroplast, which has an outer membrane and an inner membrane. The space between the outer and inner membranes is called the intermembrane space. Inside the inner membrane are flat, pancake-like structures called thylakoids. The thylakoids form stacks called grana. The liquid inside the inner membrane is called the stroma, and the space inside the thylakoids is called the thylakoid space. Figure 4. 17. The chloroplast has an outer membrane, an inner membrane, and membrane structures of thylakoids that are stacked into grana. We call the space inside the thylakoid membranes the thylakoid space. The light harvesting reactions take place in the thylakoid membranes, and sugar synthesis takes place in the fluid inside the inner membrane, which we call the stroma. Chloroplasts also have their own genome, which is contained on a single circular chromosome. The chloroplasts contain a green pigment, chlorophyll, which captures the light energy that drives the reactions of photosynthesis. Like plant cells, photosynthetic protists also have chloroplasts. Some bacteria perform photosynthesis, but their chlorophyll is not relegated to an organelle. Evolution connection. Endosymbiosis. We have mentioned that both mitochondria and chloroplasts contain DNA and ribosomes. Have you wondered why? Strong evidence points to endosymbiosis as the explanation. Symbiosis is any type of close and long-term relationship between two organisms from different species depend on each other for their survival. In some cases, both organisms benefit from the relationship. Endosymbiosis, endo equals within, is a mutually beneficial relationship in which one organism lives inside the other. Endosymbiotic relationships abound in nature. We have already mentioned that microbes that produce vitamin K live inside the human gut. This relationship is beneficial for us because we are unable to synthesize vitamin K. It is also beneficial for the microbes because they are protected from other organisms and from drying out, and they receive abundant food from the environment of the large intestine. Scientists have long noticed that bacteria, mitochondria, and chloroplasts are similar in size. We also know that bacteria have DNA and ribosomes, just like mitochondria and chloroplasts. Scientists believe that host cells and bacteria formed an endosymbiotic relationship when the host cells ingested both aerobic and autotrophic bacteria, cyanobacteria, but did not destroy them. Through many millions of years of evolution, these ingested bacteria became more specialized in their functions, with the aerobic bacteria becoming mitochondria and the autotrophic bacteria becoming chloroplasts. The central vacuole. Previously, we mentioned vacuoles as essential components of plant cells. If you look at figure 4. 8b, you will see that plant cells each have a large central vacuole that occupies most of the cell's volume. The central vacuole plays a key role in regulating the cell's concentration of water in changing environmental conditions. Have you ever noticed that if you forget to water a plant for a few days, it wilts? That's because as the water concentration in the soil becomes lower than the water concentration in the plant, water moves out of the central vacuoles and cytoplasm. As the central vacuole shrinks, it leaves the cell wall unsupported. This loss of support to the plant's cell walls results in the wilted appearance. The central vacuole also supports the cell's expansion. When the central vacuole holds more water, the cell becomes larger without having to invest considerable energy in synthesizing new cytoplasm. Learning objectives. By the end of this section, you will be able to do the following. List the components of the endomembrane system. Recognize the relationship between the endomembrane system and its functions. The endomembrane system, endo equals within, is a group of membranes and organelles, figure 4. 18 in eukaryotic cells that works together to modify, package, and transport lipids and proteins. It includes the nuclear envelope, lysosomes, and vesicles, which we have already mentioned, and the endoplasmic reticulum and Golgi apparatus, which we will cover shortly. Although not technically within the cell, the plasma membrane is included in the endomembrane system because, As you will see, it interacts with the other endomembranous organelles. The endomembrane system does not include either mitochondria or chloroplast membranes. Visual connection. The left part of this figure shows the rough ER with an integral membrane protein embedded in it. The part of the protein facing the inside of the ER has a carbohydrate attached to it. The protein is shown leaving the ER in a vesicle that fuses with the cis side of the Golgi apparatus. The Golgi apparatus consists of several layers of membranes, called cisternae. As the protein passes through the cisternae, it is further modified by the addition of more carbohydrates. Eventually, it leaves the trans face of the Golgi in a vesicle. The vesicle fuses with the cell membrane so that the carbohydrate that was on the inside of the vesicle now faces the outside of the membrane. At the same time, the contents of the vesicle are ejected from the cell. Figure 4. 18 membrane and secretory proteins are synthesized in the rough endoplasmic reticulum, RER. The RER also sometimes modifies proteins. In this illustration, a green integral membrane protein is modified by attachment of a purple carbohydrate in the ER. Vesicles with the integral protein bud from the ER and fuse with the Golgi apparatus cis face. As the protein passes along the Golgi cisternae, the addition of more carbohydrates further modifies it. After its synthesis is complete, it exits as an integral membrane protein of the vesicle that buds from the Golgi's trans face. When the vesicle fuses with the cell membrane, the protein becomes an integral portion of that cell membrane. Credit: modification of work by Magnus Manske. If a peripheral membrane protein were synthesized in the lumen, inside of the ER, would it end up on the inside or outside of the plasma membrane? The endoplasmic reticulum. The endoplasmic reticulum, ER, figure 418, is a series of interconnected membranous sacs and tubules that collectively modifies proteins and synthesizes lipids. However, these two functions take place in separate areas of the ER, the rough ER and the smooth ER, respectively. We call the ER tubules' hollow portion the lumen or cisternal space. The ER's membrane, which is a phospholipid bilayer embedded with proteins, is continuous with the nuclear envelope. Rough ER. Scientists have named the rough endoplasmic reticulum, RER, as such because the ribosomes attached to its cytoplasmic surface give it a studded appearance when viewing it through an electron microscope. Figure 4.19. In this transmission electron micrograph, the nucleus is the most prominent feature. The nucleolus is a circular, dark region inside the nucleus. A nuclear pore can be seen in the nuclear envelope that surrounds the nucleus. The rough endoplasmic reticulum surrounds the nucleus, appearing as many layers of membranes. A mitochondrion sits between the layers of the ER membrane. Figure 4.19. This transmission electron micrograph shows the rough endoplasmic reticulum and other organelles in a pancreatic cell. Credit: modification of work by Louisa Howard. Ribosomes transfer their newly synthesized proteins into the RER's lumen, where they undergo structural modifications, such as folding or acquiring side chains. These modified proteins incorporate into cellular membranes, the ER or the ER's or other organelles' membranes. The proteins can also secrete from the cell, such as protein hormones, enzymes. The RER also makes phospholipids for cellular membranes. If the phospholipids or modified proteins are not destined to stay in the RER, they will reach their destinations via transport vesicles that bud from the RER's membrane. Figure 4.18. Since the RER is engaged in modifying proteins, such as enzymes, for example, that secrete from the cell, you would be correct in assuming that the RER is abundant in cells that secrete proteins. This is the case with liver cells, for example. Smooth ER. The smooth endoplasmic reticulum, SER, is continuous with the RER, but has few or no ribosomes on its cytoplasmic surface. Figure 4.18. SER functions include synthesis of carbohydrates, lipids, and steroid hormones, detoxification of medications and poisons, and storing calcium ions. In muscle cells, a specialized SER, the sarcoplasmic reticulum, is responsible for storing calcium ions that are needed to trigger the muscle cell's coordinated contractions. Link to learning. You can watch an excellent animation of the endomembrane system here. At the end of the animation, there is a short self-assessment. Career connection. Cardiologist. Heart disease is the leading cause of death in the United States. This is primarily due to our sedentary lifestyle and our high trans fat diets. Heart failure is just one of many disabling heart conditions. Heart failure does not mean that the heart has stopped working. Rather, it means that the heart can't pump with sufficient force to transport oxygenated blood to all the vital organs. Left untreated, heart failure can lead to kidney failure and other organ failure. Cardiac muscle tissue comprises the heart's wall. Heart failure occurs when cardiac muscle cells' endoplasmic reticula do not function properly. As a result, an insufficient number of calcium ions are available to trigger a sufficient contractile force. Cardiologists, cardi equals heart. Ologist, one who studies, are doctors who specialize in treating heart diseases, including heart failure. Cardiologists can diagnose heart failure via a physical examination, results from an electrocardiogram, ECG, a test that measures the heart's electrical activity, a chest X-ray to see whether the heart is enlarged, and other tests. If the cardiologist diagnoses heart failure, they will typically prescribe appropriate medications and recommend a reduced table salt intake and a supervised exercise program. The Golgi apparatus. We have already mentioned that vesicles can bud from the ER and transport their contents elsewhere. But where do the vesicles go? Before reaching their final destination, the lipids or proteins within the transport vesicles still need sorting, packaging, and tagging so that they end up in the right place. Sorting, tagging, packaging, and distributing lipids and proteins takes place in the Golgi apparatus, also called the Golgi body, a series of flattened membranous sacs, figure 420. In this transmission electron micrograph, the Golgi apparatus appears as a stack of membranes surrounded by unnamed organelles. Figure 420, the Golgi apparatus in this white blood cell is visible as a stack of semicircular, flattened rings in the lower portion of the image. You can see several vesicles near the Golgi apparatus. Credit, modification of work by Louisa Howard. The side of the Golgi apparatus that is closer to the ER is called the cis face. The opposite side is the trans face. The transport vesicles that formed from the ER travel to the cis face, fuse with it, and empty their contents into the Golgi apparatus lumen. As the proteins and lipids travel through the Golgi, they undergo further modifications that allow them to be sorted. The most frequent modification is adding short sugar molecule chains. These newly modified proteins and lipids then tag with phosphate groups or other small molecules in order to travel to their proper destinations. Finally, the modified and tagged proteins are packaged into secretory vesicles that bud from the Golgi's trans face. While some of these vesicles deposit their contents into other cell parts where they will be used, other secretory vesicles fuse with the plasma membrane and release their contents outside the cell. In another example of form-following function, cells that engage in a great deal of secretory activity, such as salivary gland cells that secrete digestive enzymes or immune system cells that secrete antibodies, have an abundance of Golgi. In plant cells, the Golgi apparatus has the additional role of synthesizing polysaccharides, some of which are incorporated into the cell wall and some of which other cell parts use. Career connection. Geneticist. Many diseases arise from genetic mutations that prevent synthesizing critical proteins. One such disease is Lowe disease or oculocerebral syndrome because it affects the eyes, brain, and kidneys. In Lowe disease, there is a deficiency in an enzyme localized to the Golgi apparatus. Children with Lowe disease are born with cataracts, typically develop kidney disease after the first year of life, and may have intellectual disabilities. A mutation on the X chromosome causes Lowe disease. The X chromosome is one of the two human sex chromosomes, as these chromosomes determine a person's sex. Females possess two X chromosomes, while males possess one X and one Y chromosome. In females, the genes on only one of the two X chromosomes are expressed. Females who carry the Lowe disease gene on one of their X chromosomes are carriers and do not show symptoms of the disease. However, males only have one X chromosome, and the genes on this chromosome are always expressed. Therefore, males will always have Lowe disease if their X chromosome carries the Lowe disease gene. Geneticists have identified the mutated gene's location, as well as many other mutation locations that cause genetic diseases. Through prenatal testing, a pregnant person can find out if the fetus they are carrying may be afflicted with one of several genetic diseases. Geneticists analyze prenatal genetic test results and may counsel pregnant people on available options. They may also conduct genetic research that leads to new drugs or foods, or perform DNA analyses for forensic investigations. Lysosomes, in addition to their role as the digestive component and organelle recycling facility of animal cells, lysosomes are part of the endomembrane system. Lysosomes also use their hydrolytic enzymes to destroy pathogens, disease-causing organisms that might enter the cell. A good example of this occurs in macrophages, a group of white blood cells which are part of your body's immune system. In a process that scientists call phagocytosis or endocytosis, a section of the macrophage's plasma membrane invaginates, folds in, and engulfs a pathogen. The invaginated section, with the pathogen inside, then pinches itself off from the plasma membrane and becomes a vesicle. The vesicle fuses with a lysosome. The lysosome's hydrolytic enzymes then destroy the pathogen, figure 421. In this illustration, a eukaryotic cell is shown consuming a bacterium. As the bacterium is consumed, it is encapsulated in a vesicle. The vesicle fuses with a lysosome, and proteins inside the lysosome digest the bacterium. Figure 4.21. A macrophage has engulfed, phagocytized, a potentially pathogenic bacterium and then fuses with lysosomes within the cell to destroy the pathogen. Other organelles are present in the cell, but for simplicity, we do not show them. Learning objectives. By the end of this section, you will be able to do the following. Describe the cytoskeleton. Compare the roles of microfilaments, intermediate filaments, and microtubules. Compare and contrast cilia and flagella. Summarize the differences among the components of prokaryotic cells, animal cells, and plant cells. If you were to remove all the organelles from a cell, would the plasma membrane and the cytoplasm be the only components left? No. Within the cytoplasm, there would still be ions and organic molecules, plus a network of protein fibers that help maintain the cell's shape, secure some organelles in specific positions, allow cytoplasm and vesicles to move within the cell, and enable cells within multicellular organisms to move. Collectively, scientists call this network of protein fibers the cytoskeleton. There are 3 types of fibers within the cytoskeleton: microfilaments, intermediate filaments, and microtubules, figure 422. Here, we will examine each. Microfilaments line the inside of the plasma membrane, whereas microtubules radiate out from the center of the cell. Intermediate filaments form a network throughout the cell that holds organelles in place. Figure 4.22. Microfilaments thicken the cortex around the cell's inner edge. Like rubber bands, they resist tension. There are microtubules in the cell's interior where they maintain their shape by resisting compressive forces. There are intermediate filaments throughout the cell that hold organelles in place. Microfilaments. Of the 3 types of protein fibers in the cytoskeleton, microfilaments are the narrowest. They function in cellular movement, have a diameter of about 7 nm, and are comprised of 2 globular protein intertwined strands, which we call actin, figure 423. For this reason, we also call microfilaments actin filaments. This illustration shows 2 actin filaments wound together. Each actin filament is composed of many actin subunits connected together to form a chain. Figure 4.23. Intertwined actin strands comprise microfilaments. ATP powers actin to assemble its filamentous form, which serves as a track for the movement of a motor protein we call myosin. This enables actin to engage in cellular events requiring motion, such as cell division in eukaryotic cells and cytoplasmic streaming, which is the cell cytoplasm's circular movement in plant cells. Actin and myosin are plentiful in muscle cells. When your actin and myosin filaments slide past each other, your muscles contract. Microfilaments also provide some rigidity and shape to the cell. They can depolymerize, disassemble, and reform quickly, thus enabling a cell to change its shape and move. White blood cells, your body's infection-fighting cells, make good use of this ability. They can move to an infection site and phagocytize the pathogen. Link to learning. To see an example of a white blood cell in action, watch a short time-lapse video of the cell capturing two bacteria. It engulfs one and then moves on to the other. Intermediate filaments. Several strands of fibrous proteins that are wound together comprise intermediate filaments. Figure 424. Cytoskeleton elements get their name from the fact that their diameter, 8 to 10 nm, is between those of microfilaments and microtubules. This illustration shows 10 intermediate filament fibers bundled together. Figure 4. 24 intermediate filaments consist of several intertwined strands of fibrous proteins. Intermediate filaments have no role in cell movement. Their function is purely structural. They bear tension, thus maintaining the cell's shape, and anchor the nucleus and other organelles in place. Figure 4. 22 shows how intermediate filaments create a supportive scaffolding inside the cell. The intermediate filaments are the most diverse group of cytoskeletal elements. Several fibrous protein types are in the intermediate filaments. You are probably most familiar with keratin, the fibrous protein that strengthens your hair, nails, and the skin's epidermis. Microtubules. As their name implies, microtubules are small hollow tubes. Polymerized dimers of alpha-tubulin and beta-tubulin, two globular proteins, comprise the microtubule's walls, figure 425. With a diameter of about 25 nm, microtubules are cytoskeleton's widest components. They help the cell resist compression, provide a track along which vesicles move through the cell, and pull replicated chromosomes to opposite ends of a dividing cell. Like microfilaments, microtubules can disassemble and reform quickly. The left part of this figure is a molecular model of 13 polymerized dimers of alpha and beta tubulin joined together to form a hollow tube. The right part of this image shows the tubulin structure as a ring of spheres connected together. Figure 425 microtubules are hollow. Their walls consist of 13 polymerized dimers of alpha-tubulin and beta-tubulin, right image. The left image shows the tube's molecular structure. Microtubules are also the structural elements of flagella, cilia, and centrioles. The latter are the centrosome's two perpendicular bodies. In animal cells, the centrosome is the microtubule organizing center. In eukaryotic cells, flagella and cilia are quite different structurally from their counterparts in prokaryotes, as we discuss below. Flagella and cilia. The flagella singular equals flagellum, are long, hair-like structures that extend from the plasma membrane and enable an entire cell to move. For example, sperm, euglena, and some prokaryotes. When present, the cell has just one flagellum or a few flagella. However, when cilia singular equals cilium are present, many of them extend along the plasma membrane's entire surface. They are short, hair-like structures that move entire cells, such as paramecia, or substances along the cell's outer surface. For example, the cilia of cells lining the fallopian tubes that move the ovum toward the uterus, or cilia lining the cells of the respiratory tract that trap particulate matter and move it toward your nostrils. Despite their differences in length and number, flagella and cilia share a common structural arrangement of microtubules called a 9+2 array. This is an appropriate name because a single flagellum, or cilium, is made of a ring of 9 microtubule doublets, surrounding a single microtubule doublet in the center, figure 426. This transmission electron micrograph shows a cross-section of 9 microtubule doublets that form a hollow tube. Another microtubule doublet sits in the center of the tube, figure 426. This transmission electron micrograph of 2 flagella shows the microtubule's 9+2 array. 9 microtubule doublets surround a single microtubule doublet. Credit: Modification of work by Dartmouth Electron Microscope Facility, Dartmouth College. Scale bar data from Matt Russell. You have now completed a broad survey of prokaryotic and eukaryotic cell components. For a summary of cellular components in prokaryotic and eukaryotic cells, see Table 4:1 Components of prokaryotic and eukaryotic cells. Cell component function present in prokaryotes, present in animal cells, present in plant cells. Plasma membrane separates cell from external environment, controls passage of organic molecules, ions, water, oxygen, and wastes into and out of cell. Yes. Cytoplasm provides turgor pressure to plant cells as fluid inside the central vacuole. Site of many metabolic reactions. Medium in which organelles are found, yes, yes, yes. Nucleolus darkened area, within the nucleus, where ribosomal subunits are synthesized. No, yes, yes. Nucleus cell organelle, that houses DNA and direct synthesis of ribosomes and proteins. No, yes, yes. Ribosomes protein synthesis, yes, yes, yes. Mitochondria, ATP production, cellular respiration. No, yes, yes. Peroxisomes oxidize and thus break down fatty acids and amino acids, and detoxify poisons. No, yes, yes. Vesicles and vacuoles, storage and transport. Digestive function in plant cells. No, yes, yes. Centrosome, unspecified role in cell division in animal cells. Microtubule source in animal cells. No, yes, no. Lysosomes, digestion of macromolecules. Recycling of worn-out organelles. No, yes, some. Cell wall protection, structural support, and maintenance of cell shape. Yes. Primarily peptidoglycan. No, yes. Primarily cellulose. Chloroplast photosynthesis. No, no, yes. Endoplasmic reticulum modifies proteins and synthesizes lipids. No, yes, yes. Golgi apparatus modifies, sorts, tags, packages, and distributes lipids and proteins. No, yes, yes. Cytoskeleton maintains cell shape, secures organelles in specific positions, allows cytoplasm and vesicles to move within cell, and enables unicellular organisms to move independently. Yes, yes, yes. Flagella cellular locomotion. Some no, except for some plant sperm cells. Cilia cellular locomotion. Movement of particles along plasma membranes, extracellular surface, and filtration. Some no. Table 4. 1. Learning objectives. By the end of this section, you will be able to do the following. Describe the extracellular matrix. List examples of the ways that plant cells and animal cells communicate with adjacent cells. Summarize the roles of tight junctions, desmosomes, gap junctions, and plasmodesmata. You already know that tissue is a group of similar cells working together. As you might expect, if cells are to work together, they must communicate with each other, just as you need to communicate with others if you work on a group project. Let's take a look at how cells communicate with each other. Extracellular matrix of animal cells. While cells in most multicellular organisms release materials into the extracellular space, animal cells will be discussed as an example. The primary components of these materials are proteins, and the most abundant protein is collagen. Collagen fibers are interwoven with proteoglycans, which are carbohydrate-containing protein molecules. Collectively, we call these materials the extracellular matrix, figure 427. Not only does the extracellular matrix hold the cells together to form a tissue, but it also allows the cells within the tissue to communicate with each other. How can this happen? This illustration shows the plasma membrane. Embedded in the plasma membrane are integral membrane proteins called integrins. On the exterior of the cell is a vast network of collagen fibers. The fibers are attached to the integrins via a protein called fibronectin. Proteoglycan complexes also extend from the plasma membrane to the extracellular matrix. A close-up view shows that each proteoglycan complex is composed of a polysaccharide core. Proteins branch from this core, and carbohydrates branch from the proteins. The inside of the cytoplasmic membrane is lined with microfilaments of the cytoskeleton. Figure 4. 27. The extracellular matrix consists of a network of proteins and carbohydrates. Cells have protein receptors on their plasma membrane's extracellular surfaces. When a molecule within the matrix binds to the receptor, it changes the receptor's molecular structure. The receptor, in turn, changes the microfilament's conformation, positioned just inside the plasma membrane. These conformational changes induce chemical signals inside the cell that reach the nucleus and turn on or off the transcription of specific DNA sections, which affects the associated protein production, thus changing the activities within the cell. Blood clotting provides an example of the extracellular matrix's role in cell communication. When the cells lining a blood vessel are damaged, they display a protein receptor, which we call tissue factor. When tissue factor binds with another factor in the extracellular matrix, it causes platelets to adhere to the damaged blood vessel's wall, stimulates the adjacent smooth muscle cells in the blood vessel to contract, thus constricting the blood vessel, and initiates a series of steps that stimulate the platelets to produce clotting factors. Intercellular junctions. Cells can also communicate with each other via direct contact or intercellular junctions. There are differences in the ways that plant and animal and fungal cells communicate. Plasmodesmata are junctions between plant cells, whereas animal cell contacts include tight junctions, gap junctions, and desmosomes. Plasmodesmata. In general, long stretches of the plasma membranes of neighboring plant cells cannot touch one another because the cell wall that surrounds each cell separates them. Figure 4. 8. How then can a plant transfer water and other soil nutrients from its roots, through its stems, and to its leaves? Such transport uses the vascular tissues, xylem and phloem, primarily. There also exist structural modifications, which we call plasmodesmata, singular equals plasmodesma. Numerous channels that pass between adjacent plant cells' cell walls connect their cytoplasm and enable transport of materials from cell to cell, and thus throughout the plant. Figure 4. 28. This illustration shows two plant cells side by side. A gap in the cell wall, a plasmodesma, allows fluid and small molecules to pass from the cytoplasm of one cell to the cytoplasm of the other. Figure 4. 28. A plasmodesma is a channel between two adjacent plant cell walls. Plasmodesmata allow materials to pass from one plant cell's cytoplasm to an adjacent cell's cytoplasm. Tight junctions. A tight junction is a watertight seal between two adjacent animal cells. Figure 429. Proteins, predominantly two proteins called claudins and occludins, tightly hold the cells against each other. This illustration shows two cell membranes joined together by a matrix of tight junctions. Figure 429. Tight junctions form watertight connections between adjacent animal cells. Proteins create tight junction adherence. Credit: Modification of work by Mariana Ruiz Villarreal. This tight adherence prevents materials from leaking between the cells. Tight junctions are typically found in epithelial tissues that line internal organs and cavities and comprise most of the skin. For example, the tight junctions of the epithelial cells lining your urinary bladder prevent urine from leaking out into the extracellular space. Desmosomes. Also only in animal cells are desmosomes, which act like spot welds between adjacent epithelial cells. Figure 430. Cadherins, short proteins in the plasma membrane, connect to intermediate filaments to create desmosomes. The cadherins connect two adjacent cells and maintain the cells in a sheet-like formation in organs and tissues that stretch, like the skin, heart, and muscles. This illustration shows two cells fused together by a desmosome. Cadherins extend from each cell and join the two cells together. Intermediate filaments connect to cadherins on the inside of the cell. Figure 430. A desmosome forms a very strong spot weld between cells. Linking cadherins and intermediate filaments create it. Credit: Modification of work by Mariana Ruiz Villarreal. Gap junctions. Gap junctions in animal cells are like plasmodesmata in plant cells, in that they are channels between adjacent cells that allow for transporting ions, nutrients, and other substances that enable cells to communicate. Figure 431. Structurally, however, gap junctions and plasmodesmata differ. This illustration shows two cells joined together with protein pores called gap junctions that allow water and small molecules to pass through. Figure 431. A gap junction is a protein-lined pore that allows water and small molecules to pass between adjacent animal cells. Credit: Modification of work by Mariana Ruiz Villarreal. Gap junctions develop when a set of 6 proteins, connexins, in the plasma membrane arrange themselves in an elongated donut-like configuration. A connexin. When the connexin's pores, donut holes, in adjacent animal cells align, a channel between the two cells forms. Gap junctions are particularly important in cardiac muscle. The electrical signal for the muscle to contract passes efficiently through gap junctions, allowing the heart muscle cells to contract in tandem. Link to learning. To conduct a virtual microscopy lab and review the parts of a cell, work through the steps of this interactive assignment.